Hello, I'm Christine Laney. I've worked for the NSF-funded National Ecological Observatory Network, or NEON, for the last 10 years. My current role as NEON's lead of the data portal in informatics is actually on the opposite end of the data pipeline that I'll be talking about today. But I did start in data product development at NEON, and I have a keen interest in how the data get from field to publication. I have leaned heavily on several of my colleagues from different teams, and they're listed here on the first slide. If I have any questions that would be better at answering them, I will be happy to redirect to the appropriate staff. Okay, so Battelle's NEON program, it's fully funded by the National Science Foundation, and it enables large-scale ecological work by virtue of scale of data and samples, and also through the use of infrastructure and other resources. We run instruments and conduct standard protocols across 81 field sites in the US in all the major biomes of that region. As a research infrastructure, we provide free and open data and physical samples to a large community of ecologists and environmental scientists. The observatory is funded for 30 years of operations, and we've been in full operation since 2019. So these photos are just a sampling of field sites that we support. They're not static, they represent change over time. And that change can happen really rapidly, especially with major disturbances like hurricanes, fires, and also seasonal changes like ice breakup. So we have three systems of data collection and processing that we run in parallel, and I'll be talking about in depth today. The first is our automated instrument system. These are in situ instruments operating at every site 24/7. The second is our airborne observation platform, or AOP, that uses cameras, hyperspectrometers, and LiDAR to observe the land and land cover from above during peak greenness. And the third system is observational sampling, which is comprised of field crews within each domain that collect data at each site at least every 2 weeks. So NEON ingests over 5.6 billion data points per day, and the cumulative data volume is approximately 1.5 petabytes. As you can see in this graphic of NEON's data lifecycle, the data come in on the left side from our three systems into our network. They're processed and stored in Google Cloud and made available to end users through our APIs and also through our data portal, our biorepository data portal, and also partnering repositories that are domain specific. At the end, we publish more than 180 different data products. And a data product is a package of multiple data streams in one theme. So we have 5 primary themes. We have atmosphere, ecohydrology, biogeochemistry, land cover and processes, and organisms, populations, and communities, with a really wide range of data that are provided under each theme. So researchers can potentially leverage nearly any combination of these data products to study ecosystems in depth. So now I'm going to start drilling down into each system, and I'll start with our instrument system with our in situ centers. This is comprised of over 16,000 sensors across 81 sites, and it's a dynamic, perpetually self-regulating system. So the sensors were carefully chosen and placed to provide linked measurements both within and across sites. So in this left graphic, you can see, for example, that we have temperature, photosynthetically active radiation, and carbon dioxide levels being measured at different levels on a tower. We also have measurements at different soil depths, and also in where there's aquatic and terrestrial sites that are co-located. There's standardization across the sites as well. So building that infrastructure to support the instruments was really complex, and it involved design sessions and siting, permitting, and construction. We don't own any of the land that we have our sites on, so we need to continually keep our permits updated. The infrastructure itself needs to withstand really harsh conditions in a lot of areas, including major disturbances. And we have field crews that are involved in day-to-day maintenance, including sensor cleaning, sensor swap outs, calibration, and repairs. When we talk about the raw data, we're essentially talking about two different basic types of data. One is the readings, which have periodicities up to 40 hertz, especially for our eddy covariance system. We also have events which can happen at unscheduled times based on other data streaming in through the system. So a single sensor can generate more than one data stream, but all the streams contain data with a matching timestamp, then that set can be saved as a single record, which helps us save on storage and other things. Some of the data are only used at the site to inform system command and control processes to kick off events like turning on a fan or turning on a gas valve. The raw data are retained in as close as possible to the manufacturer's specifications. So on the right here is just showing a soil array at one of our sites in Utah. So it's a pretty huge area. And then on the bottom right is the interior of our instrument hut at Yellowstone with a lot of our gas cylinders for the eddy covariance system. So the data pipeline, the graphic on the left shows just the instrument systems pipeline. And I've put a orange box around the part of the system I'll be talking about. So we have the sensors connected to a computer at the site hut. The data are sent as packets into Google storage, and they can be streamed there via one of several technologies. One is a custom data logger that can collect analog or digital data. Some of the sites have Campbell data loggers, and they transmit data through LoggerNet. And then other sensors like the Lycor have their own built-in networks capabilities and can transmit data through Ethernet. The data are retained at all the sites for 60 days, and this is especially important at our most remote field sites in Alaska, particularly. And once they're ingested into GCP, they're retained as is for 2 weeks to ensure that they can be processed. Usually we only need a day for that. So like some of the other facilities that presented over the last couple of days, we use Kafka to handle the data records. As I mentioned in the last slide, some data are only needed for command and control, and they're written every second. Most of the data are published into Kafka every 8 seconds in a compressed format to reduce storage and bandwidth requirements. The sites also have metrics for monitoring processes, and these are recorded every 15 seconds and aren't run through Kafka. We use Avro schemas both for the raw data and for process data. And then we use MirrorMaker 2 to copy Kafka topics from the site computers to Google Cloud Storage. So previously, all of our data were transmitted through cellular networks or through geostationary satellite connections. But recently, we were able to install Starlink at 20 of our sites and plan to install at 5 more sites this year. This has especially been important for our most remote field sites where we were struggling with both connectivity and high costs. We're saving over $800 per month at sites that had previously used cellular connectivity. In addition, we've experienced lower latency and also increased bandwidth and reliability, especially at sites where the cellular or satellite signals were degraded or the network was overloaded. This week actually was the first time since February of 2023 where we had a morning status call and all 81 sites were online and no problems. So 16,000 sensors is pretty difficult to monitor and manage. So we can use all the software support that we can get. We have our quote unquote brain software to view how the sensors are connected into the network and what their basic statuses are. And we can use this software to install and uninstall sensors from the network. So here we're looking at all of our sensors at our Hawaii field site, and I've on the left, and I've expanded just one small section to show the individual instruments on the right side. In particular, these are wind, air temperature, net radiation sensors, and they are connected to our data loggers and then our site computer. So you can see the whole network of the instruments at any given time at any given site. We also use Grafana to build dashboards to help us rapidly visualize the status and health of sensors and processes. So this is an example of part of the dashboard for our Wind River Experimental Forest site in Washington. I just took a snapshot of this yesterday afternoon, and we can see that everything is pretty much up and that the gas valves are opening and closing as expected, and everything's running really well. So a really important part of our instrument system is our calibration, validation, and audit lab, or CalVal. So this is NEON's internal lab for annually calibrating and validating both sensors and data acquisition systems. The lab's responsible for rotating about 10,000 sensors and testing about 3,000 data acquisition systems, or DASs, per year. It ensures the performance and quality while also providing assessments of measurement uncertainty and measurement drifts over time. The in-house metrology lab consists of both indoor and outdoor facilities and fixtures, which are the stations for doing the calibration that encompasses all of the hardware, software, and controls. The lab's also developed a number of databases and apps to assist with the data management, quality controls, calibrations, data transformations, and outputs. Mobile applications have also been developed to calibrate those sensors that we can't swap out each year. So in addition to all of that, the CalVal lab also creates over 900 aquatic gas and isotopic water standards that are used in the field for traceable calibrations and validations. And finally, they also evaluate obsolete or old sensors with the new ones with the same standards to understand and quantify change over time. And this has really been useful, especially as some of our sensors right now are beyond their expected lifetime, and we need to swap them out. But we need to tell the end users basically like what are the differences, or are the data completely comparable. So that was one system. I'm going to switch over to the next, which is the Airborne Observation Platform. We also call this the AOP for our remote sensing. So this consists of 3 payloads, and each payload consists of a digital camera, an AVIRIS, which is a hyperspectrometer, and a LiDAR, which are packaged together so that they can be inserted into a small aircraft with a cutout in the belly. So this picture on the bottom left is just showing the sensors kind of poking out of the belly of the aircraft. We use 2 of the payloads continuously from March to October, flying about half of our sites each year during the window of peak greenness. We use the 3rd payload as a spare in case something goes wrong with the other 2, and also for PI requested flights through our research support services system. Those flights are done through a cost recovery, typically through NSF, NASA, or the DOE, or other granting agency. And then, so we have 40 sites planned right now for imaging in 2025. And so it's going to be yet another busy year. So at each site, we're also flying over a minimum of 100 square kilometers. And we fly lines, basically parallel lines across the entire region. And then we provide data to the end users, both by flight line and by mosaic tiles. So the AOP data pipeline is quite a bit simpler than that of the instrument system. Data are initially stored on board the aircraft, and then at night, when the field crew comes in, they're copied onto multiple hard drives using our homegrown, what we call the hotel kit. These disks are shipped to headquarters, and then the data are ingested as is into buckets in Google Cloud Storage prior to processing. We generate about 1.7 terabytes of data per flight and have about 90 flights per year, which is including the field site flights, as well as calibration flights and PI requested flights. And we collected about 157 terabytes of raw data in 2024. The waveform LiDAR data is the bulk of that volume, and it doubles in size when we extract it for the initial work. And in part of the data integrity, we use checksums on the database during that extraction process to ensure that everything's going well. So one of the key things we've had to work with over the past year is a more than doubling of the raw data that we collected compared to past years due to improved LiDAR that can operate at higher pulse frequencies. So here you see that we went from approximately 7 terabytes of LiDAR data per year to about 83 terabytes in 2024 in just the LiDAR data alone, and from about 50 or so terabytes of data total to 123 that's non-extracted. And you can actually also see in 2020 this big dip in our data collection due to the COVID pandemic. So what we can learn from this is that we can have instrument improvements happen at any time, anywhere in our system, and we need to continue evolving the cyber infrastructure to meet the science needs. It's also a big bolster to our use of cloud storage and compute, which can grow or shrink as needed. So these are just a few nice images of our final data outputs that are available to end users. So on the left, this is our Abbey Road site in Washington State. And what you're seeing are the blue dots are our field plots that are being worked on by individuals, people. And then on top of that is the raster canopy height model. And then on the right is a couple of variations on our mosaic LiDAR data at Yellowstone National Park in Wyoming. Okay, on to the third system. This is our field observations, where our quote sensors are actually people or people operating instruments. So we have about 250 to 400 field science staff at any given time, and a portion of that staffing is seasonal. And they are spread across 18 domain offices. So basically several sites per domain. These amazing people are collecting data and conducting over 50 different standardized protocols across the observatory, as well as maintaining the infrastructure in all kinds of weather conditions, including after serious disturbance events. The graphic on the top left is basically just a snapshot into the different types of biological data that we're collecting, anywhere from community composition, both in aquatic and terrestrial systems, lots of taxa. We're looking at phenology, we're looking at pathogens. We are preserving and analyzing data sequence or DNA sequences, et cetera. So the staff aren't just collecting data. They're also collecting over 400,000 physical samples every year from water, soil, and the biosphere. All of these samples have associated data that are tracked, and about 3/4 of these samples are analyzed and then discarded. But the other quarter is archived for future use by researchers. Overall, we're generating about 3 million records per year. So this is the smallest collection of our NEON data, but they're highly valuable given all of the work and labor involved on getting these samples and data. So another key aspect of the system is the many contract labs that we employ to do the further analyses of the samples. So we not only need to do quality control on the data that our staff collect, but we have a complex system to ship samples from our field offices, to transmit data, and allows labs to submit data for ingest. So our software stack that does data quality control checking on the data that comes in from the field and from the labs we just call it the parser. That is critical for both automated checking of data while also allowing field staff to correct data before the data are actually published. We push as much quality control as possible to the front end of the pipeline, but we still have a lot of humans in the loop that are in communication through different user interfaces and a ticketing system. For all of the protocols, we use the Fulcrum platform to develop applications that can be run on mobile devices or in web browsers. These applications allow for a great deal of flexibility and also structure in data recording and quality checking. They can require and constrain entries into certain fields, calculate some field values from other values, and also provide choices and limits to the field staff. Our field staff, in turn, have had a huge role in refining these apps to make them more efficient and useful. So it's a continually evolving process. To manage the relationships between the samples and their data, we use a system of sample classes and identifiers, including barcodes. Our system can link sample classes to protocols and also link parent samples with child samples. We leverage ORCIDs or ORC IDs to identify staff responsible for collecting data and samples in the field. Not only is this efficient to track internal roles of the staff, but it also provides a way for staff to obtain credit for their contributions. So we've seen a really tremendous reduction of errors in our data since we rolled out barcodes and Fulcrum maps. So on the left is the number of duplicate records before and after the use of barcodes just for our mosquito and soil data in 2018. You can just see this really big drop and very few errors after that. The chart on the right shows a total reduction in errors with the initial rollout of the apps, and that's just improved over time as well. So finally, the majority of the 100,000 samples that we retain for archival are shipped to NEON's biorepository at Arizona State University. And this is a really massive facility that's been expanding with NEON's needs for dry, wet, frozen, and cryo samples. To date, we have over 500,000 samples archived there, spanning the range from soil to water to biological. And the biological represents over 3,300 taxa of algae and plants, zooplankton and macroinvertebrates, insects, small mammals, fish, and more. We have a lot of bycatch as well. The cryo is helping preserve the microbes and genetic material in the soil samples that we have. The samples are available to the research community on request. These requests are evaluated on a case-by-case basis. But the researchers can use them destructively if they have scientific reason to do so. The biorepository assigns international generic sample numbers, or IGSNs, to every sample or specimen and provides these back to NEON, and also shares Darwin Core archive records to the Global Biodiversity Information System, or GBIF, and iDigBio and other providers for discovery. So that's it for today, but there's so much more to NEON, and I'm happy to answer questions or redirect to an appropriate team member later on. Thank you very much.